Just want to do a quick tour of the rat basal ganglia because it occurred to me that there's some major differences that you should know about. So starting with the top left section here, just quickly orient you. This is the fornix here. This is the anterior commissure right there. This is Nissl stain. So all the blue things that you see are cell bodies. And then the whiter areas are fiber tracts. So with regard to the basal ganglia, here's caudate, and here is the globus pallidus. So it's real similar to the human, and when the pallidus is where you first start seeing it is near the anterior commissure. Moving to the next section, this is still caudate, lateral to the lateral ventricle, and we have a nice globus pallidus. This is the anterior nucleus of the thalamus. This is still fornix here. All right, so moving to the next slide, we've got caudate. And this is GPE, by the way, that we're talking about. This is globus pallidus, and this is the main difference that I want to talk about. And then this is probably right here, ventrolateral nucleus of the thalamus. Moving more posteriorly, we have caudate still. This is the caudal caudate, and just for your information, we're starting to get amygdala here, which is interesting. We've lost the globus pallidus at this level, but here we have the posterior limb of the internal capsule. There. So there is no GPI in rats, and that's the major difference that I wanted to talk about. So here's caudate on the next one. And if we look really carefully here, we can see these little cell bodies right smack in the middle of the posterior limb of the internal capsule. That, in a rat, is called the entopeduncular nucleus. And that is the GPI. And there's a controversy in the field. Most people more or less ignore it. It's really hard to study because it's surrounded by white matter, very important white matter tracts that are also involved in motor activity. And so it's hard to study. Although I just did see a paper from 2017, you can start teasing out genetic subtypes of cells in there, and then use either promoters or transgenic animals to start looking at it. So that's an interesting thing. All right, so moving more posteriorly, we've already lost it, so it's very, very small. And I just want to actually, I should mention a couple other things. The striatal nigral pathway happens to be right here at the tip of the posterior limb of the internal capsule. Here we have, in the next section, we have a nice view of the subthalamic nucleus. So it's here and there. Let me get rid of it so you can see it. I'm going to outline it for you, if I can, if my drawing's good enough. Much smaller in a mouse, and so it's more difficult. So once again, posterior limb of the internal capsule, and here is still our posterior caudate, and now we're well into the amygdala in this area. Okay, and then finally, going more posterior, just to show you more parts of the basal ganglia, to orient you. This is the superior colliculus up here, periaqueductal gray. Here's the cerebral aqueduct right there. And we have, nicely enough, we have a medial geniculate nucleus. And here is a substantia nigra. We'll do that in green. Substantia nigra. This is the cerebral peduncle. Right there. So that's pretty much the entire basal ganglia.